Good afternoon, students. Thank you for joining. Um, Umaira Sayed, are you there online now? Umaira Puja. Yep. Am I audible? Am I audible, students? Shall I start the class? Puja, are you able to hear me?
Yes, ma'am.
Yeah, thank you so much. Is my microphone audible for y'all? I'm clear, right? Are you able to hear me clearly? Umaira? Umaira, are you there? Because I want to know if I'm audible to y'all so that I can take the class. So Mera has just switched on, I suppose. Pooja, shall I start? Are you able to hear me, ma? Pooja?
Yes, ma'am.
Yeah, thank you. Thank you so much. Okay, so today's class, we'll be talking about the chromosomes and karyotyping. Can you see the screen? Pooja, can you see the screen?
Yes, ma'am.
Thank you. Okay. So the topic that I've been given over here is human cytogenetics and developmental biology. Pooja, if you can, please ask others also to join the class because this is going to be a very important class and very interesting one. And I don't want anyone to miss this class. So if possible, just message them if you're all together and ask as many of them to join the class because, as I said, this is a really very interesting topic. So coming to chromosomes and karyotyping, what exactly are chromosomes and what do you mean by karyotyping is all what we're going to talk about over here. So when we are talking about the chromosomes over here, what exactly is a chromosome? A chromosome is nothing but a unit of inheritance located on the chromosome, that is the genes that are located on the chromosomes of the gametes and passed on from one generation to other, that is from parent to progeny. And it was in 1875, Strasburger, who discovered thread-like structures which appeared during cell divisions. So there was some thread-like structure that appeared during cell division. And in 1882, it was German anatomist, that is Walter Fleming, who was credited for discovering cell division in salamander larva. So that's a salamander's larva, and he was gifted or credited to study the cell divisions in salamander larva. And Walder in 1888 was the one who introduced the word chromosomes, and the number of chromosomes are fixed in each cell but varies from species to species. That is, species is humans, the canine family, the Homo sapien family, maybe the cat family, the dog family. Different characters will have different species will have different number of chromosomes over here. So the chemical constant, if you're going to see the chemical constants over here, the DNA is made up of 30 to 40% of the percentage is made up of DNA, and RNA is only 0.5 to 10%. And then there are some histones and non-histone proteins, and the proteins also make up a huge amount of the chemical constant in the nucleus over there. So when you're talking about cell divisions, there is one topic over here which has been given as mitosis and meiosis over here, or meiosis, what you call meiosis or meiosis is one and the same over here. Now, when you're talking about the cell division, before that, we will have to understand what is this, the nucleus that is made up of over here. Now, if you can see the nucleus over here, the nucleus has the nuclear pore. These are the pores from where a transcripted RNA comes, mRNA comes out through this nuclear pore. And once when they come out through this nuclear pore, the endoplasmic reticulum is attached to the nucleus around some possible nuclear pore over there. So this is one nuclear pore where the endoplasmic reticulum is attached over there, and from where the information that has been transcribed from the DNA to mRNA moves out into the endoplasmic reticulum. Now, this endoplasmic reticulum, because it is having some ribosomes on top, they are called as rough endoplasmic reticulum. And you have this nucleolus in the center of the nucleus with the chromatin, that is the condensed chromosomes that is present over here, and that's a nucleolus. And I'm sure everybody knows what is the function of a nucleolus. Nucleolus is where the RNA is going to be produced over there. So that is the nucleus. Because why we need RNA? We need RNA for the transcription and translation processes that occurs over here. Now, when this is the shape of the nucleus, inside the nucleus, you have this chromosomes. So where all those are the condensed form inside, and in the prophase stage, it is also elongated over there. But sometimes they are condensed, and sometimes they are elongated. Prophase stage, they are all completely unwound, and they are thread-like structures that you find over here. So coming to the cell division, then the one question that arises over here is why at all the cells have to undergo cell division. That is, first thing is it has to the cell has to grow in size. So for the growth, cell division is necessary. For development, also cell division is necessary, and especially for repair mechanism. Whenever there is a wound or whenever there is some problem inside the system, the cell has to repair itself. To repair again, it has to have this cell division that has to occur to repair the worn out tissues. Okay. Then the next is to facilitate exchange of materials, that is exchange of genetic information of maternal and paternal information. So for that also cell division is necessary, and also to control DNA overload. So the DNA shouldn't go extremely high, or during meiosis, the reduction division has to occur. And for that also, you need the cell division to occur over there. So when you're talking about cell division, you have to first understand the difference between prokaryotic cell division and eukaryotic cell division over here. So when you're talking about the prokaryotic cell division, in prokaryotes, of course, we all know that it has to undergo binary fission in major three steps of DNA replication, segregation, and separation. So this is what you find over here in binary fission. Okay, when you're talking about eukaryotic cell division, first thing is they have to undergo mitosis, and another one is meiosis over there. Mitosis is where the cell has to increase in number, and meiosis or meiosis where the cell has to decrease in number. That is from 46 chromosomes, they become 23 each over there, so that when these are passed on to the progeny, each progeny gets 23 from the mother and 23 from the father, and they become 46 chromosomes. So that is the eukaryotic cell divisions over there. So when you're talking about mitosis, why mitosis is important? As we all know that for the cell repair mechanism and cell growth mechanism. So for example, when you find a child being born, say after 1 or 2 months, you will find the child growing, it has grown in length. And then after say around 1 or 2 years, you will find them growing in height over there. Now all this height and repair mechanism is because of mitosis. Why? The cell is doubling itself over there in mitotic division over there. So under this division, before that, what is happening in the cell cycle? In the cell cycle, I want you all to understand. I suppose you must have done all this in your practical also, but still, I'm just going to go a little bit into the gist of it. So here, what is happening in the cell cycle? You have three to four different stages over there. What is that? First one is a G1 stage. Then you have the S phase. Then you have the G2 phase, and then you have the mitotic phase. So we all know what is this all there, but still, I'll just go through. So what is G1 phase? G stands for gap phase. So that is why the name is G. So G stands for gap phase. So here, when it is a gap phase over there, what's happening is cell growth occurs. What is a cell growth over there? So cell growth means whatever the cell organelles that are present in that particular cell. Say, for example, endoplasmic reticulum, the Golgi complex, the lysosomes, whatever is present, they will all have to duplicate itself, double itself. So that is called as cell growth over there. So they are going to double up themselves. Then it goes into the next phase called the S phase. S phase is nothing but a DNA synthesis phase over here. S phase or DNA synthesis phase, where the DNA has to duplicate itself. So that is, you have chromosomes. So what you call it as chromatid. A single chromosome is chromatid. When they duplicate, then the DNA synthesis occurs over there. So that is called as DNA synthesis over here. So all the 46 chromosomes that are going to be present, they all get duplicated itself. So duplication means doubling itself. Doubling itself means 46 becomes 92. 46 plus 46 becoming 92 over there. So that is DNA synthesis phase. Then coming to G2 phase, that is the cell growth phase again. Now, what is this growth phase over here? Now, this is the phase where checking occurs over there. What is checking over there? Your system has to again check whether every cell organelles have been doubled, or every chromosome have been duplicated itself or not. That is called checkpoint over there. So G2 phase is nothing but a checkpoint where your cell will recheck if everything is set up. Say, for example, if you're going out for some shopping or something, and all, first you will keep all your bags, your wallet, everything, money and all. So before locking the door, you will again recheck whether you have taken all those materials that is required for shopping and all. So in the same way, in the G2 phase, checkpoint is the one that checks if all the cell organelles and all the DNA have been duplicated perfectly or not. So once everything is set up, then the cell goes into the main phase called the mitotic phase, where mitosis is again divided into two types, two cells, that is mitotic cell division and cytokinesis over there. So when you're coming to the mitosis over here, as we all know, the first cell is in the interphase stage where all the cells are relaxed, the DNA is completely unwound and thread-like structures occurs over here. Now, when it undergoes mitotic phase over here, so once it comes to this mitotic phase over there, then the first stage called the prophase occurs. We all know what is a prophase. Condensation of the DNA occurs over there. As the condensation of the DNA occurs, the cell envelope begins to degenerate, nucleolus degenerates, so the cell envelope is gone over there. And then it goes into the metaphase stage where all the chromosomes, now in the late prophase stage, all the chromosomes have been condensed. You can clearly see them. And in the metaphase stage, all the cells comes and lies in the center. Meta means middle. So that is the name metaphase. So they are coming and lying in the middle of the cell over there. So that is a cell. And you can find those chromosomes in the center. And in the anaphase stage, you can find the cells, the chromosomes separating. That is here, remember, 46 has become 92. And this 92 chromosomes, that is a dyad type of a chromosomes, are present in the metaphase stage. Now this dyad is going to get separated themselves. So as they are separating, each one will have 46 chromosome over there, 46 chromosome over here. So they are going to get separated in the anaphase stage. And in a telophase stage, the cell wall occurs over there, dividing the cell into two daughter cells over this. In the late phase, you find two daughter cells. Now this is the picture that depicts onion root tip. We all must have done this in the school also. You can see different stages are presented in this one particular slide over here. You can find the interphase stage. You can clearly see the interphase stage over here. Then you also find a metaphase stage over there. Then also you find the anaphase stage. And here you can clearly see the telophase stage where two cells are getting separated. This also where the two cells getting separated. So you will have to have a good view under the microscope to find out exactly what is happening. The condensation over here, the prophase condensation, you can clearly see over there. So these are the different stages that is occurring in the mitosis. Okay, now coming to meiosis or the meiosis that is going to be occurring. The meiosis is nothing but the reduction division in which the number of chromosomes is going to be cut into half of it. That is 46 is going to become 23 each over here. So this is called as reduction division. The term meiosis was coined by Farmer and Morey in 1905. And the first this division was first studied by Van Beneden, Strasburger, and Winnewater. Why do we have to talk about the scientists? Because we have to give them the credit, because if not for these scientists, we wouldn't have been knowing still what is mitosis or what is meiosis. So a little bit of it, because these can be useful for your MCQ questions, or if you want to write any other exams, right? So you will need to have this information from here. Okay, so this meiosis was differentiated into meiosis 1 and 2, which was again found by Grigor. So this is the scientist who found out that meiosis divided into 1 and 2 over there. And American geneticist Thomas Hunt Morgan, in 1911, he observed the crossover in the Drosophila melanogaster during meiosis. So the chromosomes, they undergo crossover for genetic information to be exchanged. So who was the one, who is the scientist who discovered that? Of course, Thomas Hunt Morgan in 1911. So you might not even been understanding what had happened. Most of you all might not have been born in the year, or even I was not born in the year, but still, yeah, you can see the scientists have really done superb work on this meiosis and all the other cell divisions that is going to occur over here. Okay, coming to the stages over there. Now, what is the stage over here in the prophase of meiosis 1 and 2 over here? Prophase 1 is again divided. Here, I want you all to understand. Now, this is the phase that they will be asking you all for the examination point of view. Why? Because in meiosis, meiosis 1 and 2 division, meiosis 1 is very, very important. Why? Because the prophase stage has these type of different stages that is being divided, and then it goes into metaphase, anaphase, telophase. Now, thankfully, mitosis and meiosis have the same name over there, that is prophase, metaphase, anaphase, and telophase. Now, only difference is what is happening in the mitosis and what is happening in the meiosis is what you'll have to understand. And what I want you all to pay attention is what is happening in the prophase stage of meiosis 1. Now, when we are writing 1 over here, that means I'm talking about meiosis 1. And if you're writing 2 over there, I'm talking about meiosis 2 over here. So this is how you're going to differentiate between meiosis 1 and meiosis 2 over here. Now, when you're talking about meiosis 1, the prophase stage is again divided into leptoteenage, zygotene, pachytene, diplotene, diakinesis. I'm sure you must have done all this in your practicals, but nevertheless, because we have this in the theory, I'll just go through what is important in this point of view over here. Now, what exactly is happening in this stage? Now, this is a very, very important phase of prophase 1. Why? Because the recombination of the genes occurs over here between homologous chromosomes. The homologous recombination, that is, the sharing of information between the maternal and paternal gene occurs in this prophase 1. Now, why is this given so much of importance? Is because if there was no recombination during prophase 1, then we might not have had this many combinations of humans in this world. Now, for example, we say that no two person are alike. No two person fingertips, finger mark is the same over there. Fingerprinting is different from each one person to another person. Now, given the fact that human population, there's so much of human population over there. Now, in that population, you say that no two person are alike. Now, how is this possible? Possible because we have this homologous recombination. Now, what is the amount of recombination that you can get? Given the fact that we have 46 number of chromosomes, all the 46 number of chromosomes has to undergo recombination between these homologous chromosomes. So what will happen? Imagine that we will be getting somewhere around 70 trillion combinations. Now, in the 70 trillion combinations, you can definitely expect, not in this century, even if the coming century, for ages and ages, no two person is going to be alike. So that is why government takes your fingerprint, because no two person is, and also they take the eye printing also. Why? Because no two persons eye lenses are going to be the same. So that is the beauty of meiosis, the meiosis 1, prophase 1, all these stages over there. So what is exactly happening in this homologous chromosome is what we are going to talk about over there. Now, coming to the stages over there, the stages can be divided into, as I told you, leptotene, zygotene, pachytene, diplotene, and diakinesis. So as the picture depicts over there, I will show you all slide by slide what is happening over there. Now, so in the leptotene stages over here, I'm sorry, this is somewhere else, this shouldn't come here. Now coming to leptoteen stage over there, the first stage of prophase 1 is the leptoteen stage, as you said. Lepto means thin, teen means thread. Thin thread is what you're going to talk about as leptoteen. So during this stage, individual chromosomes begin to condense and long threads into long threads within the nucleus. So this is a nucleus and the long threads are going to start to condense. So however, two sister chromatids are still tightly bound as they are indistinguishable from one another. Two sister chromatids. Now what is this sister chromatids? Now what I want you all to understand is when you're talking about the chromosomes, the chromatid, when it is not in the S phase, it is a chromatid. But when during S phase, this chromatid will duplicate itself. Now that becomes cystochromatids. Now these cystochromatids are tightly bound and they are not distinguishable from one another. So they are tightly bound like this. You cannot see them like that. You cannot see them like that. They are tightly bound. So this feels that you have only single chromosome over there. So this is leptoteen stage. Okay. Now coming to zygotine stage over there. So what is a zygotine stage over here? Zygotine is also known as zygonema, called paired threads. Zygo is paired threads. Now, what is happening over here? Pairing of the chromosomes occur over here, and this is known as zygotine stage. Pairing of the chromosome or synapses occurs over here. What is synapses? Pairing of the chromosome occurs over there. And what is happening over here as the pairing of the homologous chromosomes occurs? What do you mean by homologous chromosome over here? When I'm talking about chromosome 1 of mother or maternal chromosome, I'm talking about the chromosome 1 of paternal. So chromosome 1 of maternal, chromosome 1 of paternal. Now this chromosome 1 and chromosome 1 from each parent is called as homologous chromosome. Homo is same. Homologous chromosomes. Now, synapses occurs over here. That is, chromosome 1 will pair only with the chromosome 1 of maternal paternal. Chromosome 1 will not pair with a chromosome of chromosome number 2. So, chromosome number 2 is different. So, chromosome number 1 of maternal, chromosome number 1 of paternal, same is called as homologous chromosomes over here. Now, the pairing occurs between these two homologous chromosomes, that is called as zygotine stage. Pairing of the chromosome or synapses occurs over here. Now, what is a synapses over here? Now, chromosome 1 of maternal, chromosome 1 of paternal over here, these two join together, and when they come together, they should not move away. So, why? Because in that nucleus, you can imagine nucleus is such a small, tiny one, this 2 nanometers diameter, and inside that all these chromosomes are present. Now what, when you join all the chromosomes together one by one, what is the size of the chromosome? You will get around 1.8 meters long. Now this 1.8 meter long thread has to go and settle inside this 2 nanometer diameter of nucleus over there. So chances of it moving here and there, or chances of other chromosome coming and joining together will occur. So when the homologous chromosome come close by to each other, they identify each other, and you will find that the synaptonymal complex is formed over here. You can see the synaptonymal complex. This doesn't allow them to move away. So that is the beauty of zygotine over there. Now coming to pachytene. Now once pachytene is occurring, pachytene is nothing but crossing over is called as pachytene stage over here. So you can find the chromosome crossing over. Say for example, this is a maternal chromosome, that's a paternal chromosome, and there is a crossing over that is occurring over here. Why? The sharing of information has to occur in the pachytene stage, and so this is called as pachytene stage, where crossing over occurs over here. So this is the crossing over. Another name for crossing over is called chiasma formation. So that is a chiasma formation where, so crossing over. So this is called as crossing over or the chiasma formation, where information is exchanged between the two chromosomes, that is maternal, paternal chromosome of homologous chromosome. I want you all to again and again remember, there is a crossing over only between homologous chromosome. That is, chromosome number 2 will pair with chromosome number 2 of the maternal paternal. Chromosome number 10, for example, is going to pair up with the chromosome number 10 of maternal paternal. A 10 will never pair with 11, or a 12 will never pair with 20. So 20 will pair only with 20. 10 will pair only with 10. So that is the homologous chromosome that we are talking about. And here the crossing over occurs. Now this is the beauty over here of crossing over. Why? Because of the exchange of the chromosome of maternal character and the paternal character, if this chromosome is going to be inherited by the child, now you will say that the once a child is born, everyone must have experienced this particular point over there, where people say that when a child is born, this resembles like the mother, same face cut, or this resembles like the father, same eyes, same hairstyle. Why? Because the exchange of the genes is occurring at this point over here, and that is the reason why when a child is born, we say that the child resembles like a mother, or child resembles like a father, or child resembles like Attamma, Menata. All those different characters is because of this particular stage of packaging where information is being exchanged, and that is why no two persons are alike. 70 trillion. I'm talking only about one chromosome over here. So here in this picture, you can see only one or two chromosomes getting exchanged. Imagine all the 46 going to get exchanged like that. Okay. Now coming to diplotin stage, diplo means two thread. So here, after the crossing over has occurred, slowly the threads moves away. So as they are moving away, they look like two threads over here. So very clearly you can find two threads over there. So in this point over here, they are very close over here. You can see that they're very close. You cannot identify the crossing over that's occurring. But in a diplotin stage, you will find that they're getting separated, something like that. So you can see them, they are getting separated. This is one homologous chromosome. This is one homologous chromosome over here. The crossing over that has occurred between the exchange of the information that is occurring, and they are moving away. Now, as they are moving away, they look like two threads. So the name diplotin stage over there. Okay, diakinesis is completely, they have moved apart, and now they are going to look like two threads over there. And after the movement has occurred over here, you can clearly see in this particular picture over there. Now, if you can see these two threads over there, now this blue color one has taken over an information of the red one over there, and this red one has taken the information of the blue one over there. So exchange of chromosomes has occurred. Same thing occurred at the tip over here. Now the same thing occurred. Now because this chromosome is a smaller one, maybe this chromosome must have had only one crossover, whereas this chromosome must have had two crossovers. One crossover over there, one crossover here, two crossovers. And this, because they are very small, and these are submetacentric. I'll talk about what is submetacentric. Now, the crossing over occurred only in one area over there. So exchange of information occurred only at one point over there. So diakinesis, the word, that is, they have further moved away. And now you can clearly see the dyads separated over there. And the chiasma also a little bit very clear over there. Chiasma formation is also very clear in diakinesis. So that was about prophase 1, where we talked about leptotin, zygotin, pachytin, and diplotin over there. Now, exactly, if you want to remember the stages over there, I will just give you one example of how you have to remember these stages, because sometimes when you're writing the exam, you will have a tension and you'll have a doubt whether pachytin comes first or diplotin comes second, or zygotin comes first. There's a lot of confusion over there that is going to occur over there. So what you can do is, you'll have to take down only the first letter of each stages over there. L, Z, P, and D over there. So you can clearly see in this picture, L, Z, P, D, and D over here. So what you can remember as L stands for Lata, Z stands for Zara, P stands for Pani, D for they. Lata, Zara, Pani, they. If you remember that, you'll be able to write prophase 1 very clearly, because in your book, he has mentioned describe prophase 1 in detail. So when you're going to describe prophase 1 in detail, you will have to talk about all these leptotin, zygotin, pachytin, diplotin, diakinesis. Now, because I've given you all these points over there, you can just go through these points and you can write these points for your examination point of view. Okay. Coming to what is happening. So prophase 1 is over. Then it goes into the metaphase stage, where all the cells are going to be aligned after the exchange of the information. All the chromosomes are going to get aligned in the center, and they're going to get separated. How they're going to get separated? With the spindle fibers, the chromosome are going to get separated, and then they go into telophase, where two cells are formed over there. Now, still, these are not completely divided. Now, if you remember, from 96, they have become only 40, 92, they have become 42, 46 over here. Now, the 46 has to become 23. So, they have to undergo the next division, that is meiosis 2 division. So, in the meiosis 2 division, the same stages occur: prophase 2, metaphase 2, anaphase 2, and telophase 2. So, what is happening? The prophase 2 is a very short duration. Nuclear membrane and the nucleolus disappears fast, and then the spindle fiber is formed over there. The spindle fibers are ready over there, and they get attached over there. As the spindle fibers, centrioles forms a spindle fiber. The spindle fibers goes and gets attached to the centromeres, the kinetochores that are present near the centromere. They go and get attached, and the dyads. Now this is called as a dyad, right? And these dyads are going to get separated over here. And in the anaphase, the separation occurs over here. And as the separation occurs over here, you can see now. Now, what I want you all to understand is in the anaphase 1, so here, if you can see in the anaphase 1, they are still in a dyad form. They are not N form, 2N. They are still in 2N stage. They have to become N stage. So here, this is becoming N stage over here. 2N has become N. That is, diploid has become haploid. So you can see haploid over here. So now when this gets separated, each cell is going to have, so you can see over here, each cell is going to have one single chromatid from each parents over there. And now each gamete is going to have 23 chromosomes each. So this will have 23, this one will have 23. Now in mitosis, the daughter cells will have 46, 46. But here in meiosis, because a cell reduction is occurring, now each cell is going to have 23 and 23. So that is the beauty of meiosis 1 and meiosis 2 over here. So this has become haploid cells over here from diploid. So here you can see the prophase 2, they're going to get separated to give haploid number. So what is the significance of this meiosis in the gametogenesis? Now we are the cells, the chromosomes are divided into two types. We all know that is, one is somatic cells, and the other one is the allosomes over here. Somatic cells are the cells of the body, that is from the head, tip of the head till the tip of the body is the somatic cells. And allosomes, or the germ cells, are the sex chromosomes, which is going to be the reproductive cells. Now, the reproductive cells are the ones which will undergo this meiosis 1 and 2 over here. So what is the significance over here? When you're talking about the meiosis over here, when you're talking about the spermatogenesis, the spermatogenesis undergoes first mitotic division. So mitotic division, remember, the 46 has become 92 over here. So they undergo, they become primary spermatocytes. Now this primary spermatocytes will undergo this one division to form secondary spermatocyte. In the secondary spermatocytes, this 92 has become 46, 46 over here. Now this 46 has to become 23, 23. So this secondary spermatocyte will undergo meiosis 2 over here. And after the meiosis 2, in spermatocytes, because they are having X and the Y genes, the chromosomes that are present over there, X and Y chromosome, each cell will now have, one will have 23X, one will have 23Y, 23X and 23Y. So that each sperm will either be an X chromosome or it will be having one Y chromosome. So one mother cell is going to give 4 sperms over here in spermatogenesis. When you're going to talk about the oogenesis, the oogenesis again has to undergo the same mitotic division, which will become primary oocyte. Primary oocyte Students, can you all hear me now? For a short period, I lost my connection. Pooja, can you hear me? Shravani, are you all there, students?
Yes, ma'am.
Sorry, because there was some slight problem in my connection. So, okay. So I was talking about oogenesis over here. So once the first polar body is released, the first polar body dies, and then you have the second polar, the second oocyte over there. The secondary oocyte undergoes meiosis to completes the cell division, and what you get is a complete ovum over here, and you have another secondary polar body. The second polar body also dies, and what you're left out with only one ovum over here. So under oogenesis, during cell division, one mother cell will give rise to only one ovum, and not like the spermatogenesis where one cell is going to give rise to 4 sperms over there. Okay, so what happens in this ovum, and I suppose we'll talk about this bar body later on over here, but here you can just find out you have a pink color and a green color over here. So when a chromosome is being dyed with a fluorescence probe, you can find the X and Y colors that are present over here. And if it is an female, you have only 2 X's over there. That is 2 pink colors that is being found over there. That's only for you all to understand how they look like. So coming to the chromosomes and the types of the chromosomes that you're talking about over here. So the chromosomes are of 2 types over here. So what are they? As I told you all, autosomes and sex chromosomes are present over here. So when the autosome, what exactly is an autosome? That is the character other than the sex character. That is the somatic character from the tip of the head till the tip of the body is the somatic characters that is found. That is those who's called as autosomes. And the sex chromosomes are nothing but the X and the Y chromosomes are called as the sex chromosome. This is only to differentiate between a male and a female. We call them as sex chromosome, X and Y chromosome. So the humans mostly have XX means female, male means heterogametes, that is X and Y. Homogametes, XX, heterogametic is X and Y over here. If a child is having 2 different sex chromosome, that is X and Y chromosome, then it is called as male. But when you're talking about the birds over here, the birds are a little bit different over here. What is it? The female is heterozygotic. So ZW, so if it is a ZW, then it's a female. And if it is ZZ, then it is a male over there. So that's just a little bit of difference that you find in this male and the Homo sapiens and the birds over here. Now coming to chromosome number. Now what is this chromosome number? Haploid, diploid, triploid, and tetraploid. So the chromosomes over here, haploid is if it is only 1, it is called as haploid. If it is 2, then it is diploid, 3, triploid, 4, tetraploid. Now, in humans, it is a diploid number of chromosomes. What we have is a diploid number of chromosomes. So what do you mean by a diploid number of chromosomes? 1 chromosome from the paternal side and 1 chromosome from the maternal side is 2 chromosomes that you inherit, one each 23 chromosomes from each parent. And that is why humans have diploid number of chromosomes. Now, who is going to have this triploid and tetraploid? That is each gamete having 3 times over there. So if this is chromosome number 1, there are 3 chromosomes over here. If this is chromosome number 2, there are 3 chromosomes over here. Now, what is this over here? Now, this type of a chromosome, triploid and tetraploid, you find only in the plants and not in animals over there. Or mostly in animals, you don't find them. But this type is found only in the plants. We're not talking about plants, so we'll just not talk into the detail about that. Now, a little bit of information about this different types of chromosomes. So here you can find species, human species have 2n. So as I told you all, the animals all have 2n, that is diploid number of chromosomes. So here you have diploid number of chromosomes, one from the mother and one from the father over there. So if you can see humans at 46, pea plant at 14, chromosome number is 14 over there. If you're going to talk about the cat, 38, cattle at 60, pig at 38. So these are different numbers that you can find over in ostrich at 80 over here, elephant at 46 over there, and Drosophila or the fruit fly at 8 over there. And say for lion, it's at 38. So each one will have different number of chromosomes. That is what we find over here. Even though housefly over here has 12, for example, Drosophila 8, the fruit fly, they are so close to humans because some of the genome that is found in the fruit fly resembles that of the humans, and that is why there is a lot of research that is going on the Drosophila, which mimics the human disease over there, even though the chromosome is less over there. Okay, now coming to what is chromatid and what is chromosome. So when you're talking about the genetics part of it, you will have to understand what is a chromosome. When you're saying chromosome, what is chromosome? And when you're saying chromatid, what is chromatid? Now, if it is in a single form of year, that is 23. If you remember, that is what I was talking about. 23 from mother, 23 from the father. This is the chromatid that we are talking about when they have not duplicated. It is called as chromatid. Once they duplicate, they become chromosomes. How duplication occurs? In which states the duplication of each chromosome occurs? If you remember the cell cycle, the duplication occurs in the S phase, synthesis phase, DNA synthesis phase. This duplication occurs over there. DNA duplication occurs over here, and this too becomes cystochromatids. Now, because this is going to have the same copy over there, like this, same copy over here, now that is a cystochromatid. Now this is also going to have the same copy over there. This is also a cystochromatid. Now if these two chromosomes belongs to same number, that is if I'm talking about chromosome number 5. Say for example, if this is chromosome number 5 and this is also chromosome number 5 coming from each parent over there. Now because these two chromosomes are chromosome number 5, then you call it as homologous chromosome. But if this chromosome is chromosome number 5 and if this chromosome could have been chromosome number 7, 5, 7 becomes non-homologous chromosome. If you remember when I was talking about the crossing over occurs only between homologous chromosome. That is, if these two are the same chromosome, homologous chromosome, and if these two are different chromosome, then they become non-homologous chromosome. Crossing over does not occur between non-homologous chromosomes. Okay, so how do you differentiate these? The one that is going to attach this chromosome together is the center part, that is called the centromere. So the centromere is attached in the center, which keeps these two cystochromatids together. Okay, coming to structure. Now this is very important. Chromosome structure over here. So when you're talking about the chromosome structure over here, the chromosome has two arms over there. Now, this is one arm over there. This is the another arm over here. These arms are divided in the center with a centromere. So that's a centromere that is present in the center. The spelling is wrong over there. So that's the spelling centromere. That's a centromere over there. The arm that is above the centromere is called as a P arm. The arm below the centromere is called the Q arm. Now you will have a doubt. Now, what if it is upside down? So if when it is going to be upside down, how do you differentiate whether it is a P arm or a Q arm over there? Now, how you are going to differentiate that? Now, this depending on the position of the centromere, if the centromere is dividing the chromatid in two equal arms, then both P arm and Q arm. But if the centromere is going to divide the arms in unequal arms, always the P arm is smaller and the Q arm is longer over there. So if you can see in this picture over here, now this P arm is smaller and Q arm is longer. So the one that is longer, that is called as a Q arm. The one that is smaller, that is called as a P arm. P stands for petite. Petite means delicate, small. So the name P arm. Then why is this called the Q arm? Simply that ABCD after P comes Q, so Q arm. So that's all. Nothing more than that over there. Now, depending on the constrictions of here, now the centromere constricts the chromatid at one point of here in the center. There is a constriction that is going to be, it's pressed a little bit. That's called as constriction. Now, the centromere is called as a primary constriction. That's a centromere. Primary constriction. That's a primary constriction. Sometimes there is a constriction at the either end of the P arm or the Q arm over there. So there's a constriction over here. There's a constriction over there. Now this type of a constriction is called as secondary constriction. And at the secondary constriction, if the constriction is at the P arm, if the constriction is present at the P arm, the small short arm that is formed on top, now that is called as a satellite over there. So that is a satellite. not the satellite of the universe where we send to the Earth or Moon over there, not that satellite. The satellite that is present on top of the P arm over there, somewhere over there, and that is called as a satellite over there. What is the function of a satellite? We'll talk about that. Now, the covering of the chromatid is called as a pellicle. The chromosome is covered by a pellicle, and inside you have the matrix that is present over there, and the DNA that is present inside is called as the chromonimata. Chromonimata. Chromo means chromosome. Nimata is thread. Thread-like structure is called as the chromosome. So the name chromonimata. So that is the chromosome differentiated as pellicle, matrix, and chromonimata. Now we also know what is a P arm, Q arm, and a centromere, and these are the bands that are found. What is the importance of the bands? We'll be talking about that in the next in the following structures. Now coming to the chromosome, chromosomes are of 4 types over there. Depending on the position of the centromere, you're going to divide the chromosome into 4 different types. What are the 4 different types of here? So these are metacentric, submetacentric, acrocentric, and telocentric chromosomes. So what is this metacentric, submetacentric, acrocentric, and telocentric? Now, for example, if the centromere is present in the center of the chromosomes, dividing the arms into 2 equal arms, then it is called as meta. So the M stands for meta, meta stands for middle. So if the chromosome is divided into 2 equal arms, that is called as metacentric. If the chromosome divides the arms into 2 unequal arms, one short arm, that is a P arm is short, Q arm is long, then it is called as submetacentric. And if you have an acrocentric, the centromere is dividing the arms into very small P arm and a very long Q arm, then it is called as acrocentric. And in the telocentric, if the centromere is dividing the 2 arms into 2 unequal, that is completely absence of P arm and only Q arm is present, then you call it as a telocentric. Telo means the presence of centromere at the telomerase end of the chromosome. What is the telomerase end? The ends of these chromosomes are having telomers over there. Cap-like structure is found over here. And because they are present at the telomerase site, then this is called as telocentric chromosome. Now, this telocentric chromosome is not found in humans. In humans, we have only these 3 types. You'll have to only remember metacentric, submetacentric, and acrocentric chromosomes. So that is the definition of different types of chromosomes that you find in humans over there. Metacentric, when the arms are equal and the centromere is positioned in the center, you call it as metacentric. In submetacentric, if you have a small P arm and a long Q arm, then it is called submetacentric. And in acrocentric, very small P arm and very long Q arm, you call it as acrocentric. So our chromosome is divided into 3 different types. And this will help us in understanding the karyotype of the chromosome. So what is the karyotyping of the chromosome? You are typing, you're giving a name to the particular chromosome, that is called as karyotype. What example is a karyotype over here? How you differentiate between a male and a female karyotype? That is, if the chromosome has 2 X chromosome, then you call it as female. And if the chromosome have 1 X and 1 Y, you call it as male. So that is called karyotyping. You're giving a name to that particular chromosome. So that is called as karyotyping. And in this karyotyping, this will come into help for us. So how it is going to come into help for us? Now, as I told you, you already know what a secondary constriction is. So you can see over here, the P arm is getting constricted at the ends over here. So at one end, that is on the P arm, the constriction is present over here, and you have a satellite over there. And this satellite structure is called as NOR region, nucleolar organizing region. So that's called as nucleolar organizing region. And what is the function of a nucleolar organizing region or nucleolus organizing region? So this region is the one that is going to give rise to nucleolus in the nucleus. Now, as I told in the beginning, what is the function of a nucleolus? Nucleolus is the one that gives rise to RNAs, right? So that is the function of nucleolus. So this is the one that is going to give rise to rRNA and tRNA, nucleolar organizing region, that is going to transcribe the rRNA and tRNA. So that is the importance of NOR. So remember that over there. So you have the centromere. The centromere is having kinetochore. The kinetochore is the one that is going to help in identifying the spindle fibers and going to get attached to the centromere, and then they help in anaphase and telophase stages over there. Now, telomeres. What is this telomeres? I was talking about the telocentric over here. Now, telomeres are the one that is the extreme ends of the chromosomes over here. The extreme ends of the chromosome has a cap-like structure, and this cap-like structure is called as telomeres or telomere center. Telomerase are the one that helps in DNA repair mechanism. So this is the one that helps in DNA repair mechanism. As we are aging, you must have seen that repair mechanism slows down. And when it is a young child, DNA repair mechanism is at its peak. So that is, for example, if a child has a say a fracture in the bone, they will be able to get up in a, say in 15 days to 1 month, they get up and run. But if it is a grandmother who's going to fall, you can imagine the time that it will take for them to repair. Why? Because as we age, the telomerase is also becoming shorter by the age. Why? Because we have used up all these telomeres as we are aging, the repair mechanism has occurred, and hence the telomere shortens, and that's the reason why repair mechanism also shortens. So telomere is very, very important for the chromosomes, and there are a lot of research that is going on the telomerase. Why? Because humans are so very greedy, like in a sense, why greedy in a sense? We don't want to age at all. Everyone wants to stay young as they were in their 20s and 30s over there. Now, scientists are doing this research over here, how to keep a person young eternally. And they're doing a lot of work on this telomeres. And if anybody of you all are interested in doing some research work, so please take up this research on telomeres. Maybe who knows who's going to get a Nobel Prize over there if you're going to break through what exactly it is over there. Okay, coming to the next composition of the chromosomes over here. So we have understood centromeres, we have understood the different types of chromosomes, and we have understood the telomeres over there. Now coming to the compositions, now when you're talking about the chromosome, when you stain them, you will have this band-like structures, dark band and light band over there. Now, dark band, light band are the one that is going to have a specific function over there. That is why we have this bands that are present over there. Now, this bands predicts whether the region is heterochromatin or euchromatin. You must have heard this word, heterochromatin and euchromatin. If the bands are very dark in color, if say for example, if you're going to take this band over here, if the bands are very dark over there, you can see those dark color ones over there, then these are called as heterochromatin. And if they are a little bit lighter in shade, then you call it as euchromatin. So what is the function of a heterochromatin and a euchromatin? Now, whenever you need some proteins to be transcribed or transcription and translation has to occur, your DNA has an information. Say, for example, we all must have had a food just now, and we need digestive juices to digest whatever food that we ate now. Now, for that, what your body has to do? Your body has to produce that enzymes. Okay. Now, who is going to tell what type of enzyme has to be produced? DNA has to tell. Now, who is going to give the information? The DNA has to go give the information so that the mRNA copies that information, comes out, and rRNA and tRNA is going to transcribe them to produce that particular protein of interest for us. Now, which part of the DNA? Now, euchromatin is the one that is going to give that information and not the heterochromatin. Heterochromatins are tightly condensed. They do not give information for translation or transcription. Translation is not happening in the heterochromatin. The translation occurs only in the euchromatin region. So this region where the DNA is loose over here, they have the information over there, and that is the one that is going to give us. That is the difference between euchromatin and heterochromatin. Heterochromatin are very, very, very secret. They keep their DNA very secretly preserved inside. They do not give the information. Who is the one that is going to give the information? The region that is euchromatin region. So this light region over there is the one that is ready for us to take the information, and the dark bands over here are the heterochromatins, which does not allow the information to be given. So that is the difference between euchromatin and heterochromatin. But in this heterochromatin, that is a tightly placed one. It is again made up of two types. What are they? Constitutive and facultative. Now, constitutive cells, they are identical, and they will never give information at all. That is constitutive. Facultative is during emergency, when the euchromatin doesn't have that information with it. It is a facultative of the heterochromatin that will say, okay, I will give you the information. So that is facultative heterochromatin. That is heterochromatin divided into two types, constitutive and facultative. Constitutive will never give information. Facultative, only during emergency situation, they will give information to us. Otherwise, usually it is the euchromatin that is going to give us the information over there. Now, this particular chromosome that is depicted over here, I want you all to understand how the patterning, bands pattern, and how we talk about in this particular gene is, how you give the name for each gene, banding patterns of the genes on the bands over here. Now, you can see this is the P arm. Because why I'm saying this is as a P arm, this is a short arm, and this is a long arm, Q arm over there. The P arm is again divided into two regions, region 1 and 2. So if you divide this P arm into two, region 1 over there, region 2 over there. And again, region 1 is divided into 1, 1, that is 1, 2, 3, 4. And region 2 is again divided into 1, 2, 3. So it is going from bottom side, 1, 2, 3, 4, 1, 2, 3. So if there is a problem, say for example, if I'm going to talk about this particular gene, how do I depict that? P arm at the first region over there, the first region at this point over there, this point 2. So P 11.2. What is 11.2? So if I'm going to divide this into two parts, the first region is 1. So 1. Under 1, again subdivision 1. And a subdivision 1, I'm talking about this 2 over there. So this point over there. So P 11.2. So for example, if I'm going to talk about P 22.2 over there, so this is P region, second point, second subdivision, and this point over there, P 22.2. So you will have to understand this in detail for y'all, just to understand how we give the nomenclature of each genes over there. I will come and talk about this later on. I want you all to understand first exactly how the ultrastructure of the chromosome is. So there is one topic over there, ultrastructure of the chromosome, how the chromosomes are packed up. That is, I told you all, if you open all the chromosome in lengthwise, you will find it is almost 1.8 meters. So how is this 1.8 meter long cloth is condensed so small? How they are condensed? Because they are highly coiled. How they are highly coiled? Now, if you can see over here, your chromosome is having a nuclear scaffold. A nuclear scaffold, further when you open it, you will find a solenoid model. And further when you open it, you will have a nucleosomal model. And once the nucleosome model is again opening up, you can clearly see the DNA double helical structure. Now, if I'm going to come back like that, now this double DNA helical structure is wound so closely on the nucleosomal model over there, and this one becomes a solenoid, and this solenoid becomes nuclear scaffold, and further condensation becomes the chromosome over there. How this is being done over there. We'll talk a little bit about the solenoid model over there, model which was proposed by Kornberg and Thomas in 1974. What is happening is this chromosome gets wound around the nucleosomes. Now, what is the nucleosome over there? The histone proteins that we are talking about, the DNA and the histone core proteins over there. Now, the histone proteins over here are made up of H1, H2A, H2B, H3, and H4. Now, this solenoid model is going to have H2A, H2B, H3, and H4. Exactly, if I'm going to talk about this. See, so this is the point over there, the nucleosome over there. Now, this is the histone protein over here. The histone protein is made up of H2A, H2B, H3, and H4. The same thing is again copied below, and that becomes a octamer model. So 4 on top, 4 below them, that becomes the octamer, 8 histone proteins that are present over here. Now, around this is wound your DNA two times, that is 1.65 turns per nucleosome, the histone protein around them. So 1.6 or 1.68 or 1.75, that is close to 2 turns. So it is close to 2 turns. So a 2 turn is done by your DNA around. Now, once the DNA is been turned around, now what happens sometimes, for example, if you take a ball and put a thread around, and if you leave it, it will come out. But here in this DNA, it doesn't come out. Why? Because the histone protein H1, I told you all H1 also, this H1 acts as a plaster that doesn't allow the DNA to unwind. So once it is wound, H1 goes and sticks to them as a plaster. So like that, they keep on packing themselves in a solenoid model like this, and each solenoid again supercoils itself, and then that supercoil becomes a condensed form of the chromosome. So that is how the model is depicted over here. So this model is depicted as nucleosomal model, where you have the octamer, that is the histone protein. All these histone protein again forms a tetramer or condensation of the nuclear scaffold. So each per round, it will have 6 solenoid models over there. This is one solenoid, 6 times over there. And this becomes condensed, highly condensed coil, and then they become a chromosome. So if you remember earlier and all, we used to have this telephone wire. Now, telephone wire, sometimes it gets coiled, supercoiled so much that it is very difficult for you all to separate them. Then you'll have to slowly uncoil them. So that is how this supercoiling occurs over here, and the chromatin, the chromosome becomes highly condensed. So that is how the structure is formed over here with the histone proteins. Now, the histone protein, what is the charge of the histone proteins of that? The charge of the histone protein is positive. Why? Because histone proteins, they have this lysine and arginine, making them positive. Whereas a DNA has phosphate group, the phosphate groups are negative. So negative and positive attached together. That is why they get tightly coiled among each other. So DNA is tightly coiled around the histone proteins over there. Okay, coming to karyotyping. So as of now, I suppose you have understood what exactly I'm talking about. If there's any questions, you can keep interrupting me, because there is no session that is complete without we having a proper interaction between the student and the lecturer. So if there is any point that I have to keep making you all understand, please let me know. You can interrupt me, you can ask me the questions so that I can go into the next topic. Okay, as of now, I don't find anyone asking me questions, so I will continue with karyotyping over here. So what is karyotyping? As I told you, karyotyping is you're giving some name to the chromosomes, and that is why you call it as karyotyping over here. Now, as the picture depicts over here, you can find chromosome number 1, 2, 3, 4, 5 up till 22. And then all these chromosome number 1 to 22 are autosomes. And chromosome number 23 is called as a sex chromosome. It can be an X chromosome or a Y chromosome over here. Now, you can also see that there is a numbering that is given to each chromosomes. Now, how is this numbering given over here? Now, chromosome number 1 is the tallest among all the other chromosomes, and chromosome number 22 is the shortest. Now, when you arrange this chromosome, you can arrange them in a descending order from the tallest to the shortest. So this is the one that gives the name to each chromosome, chromosome number 1, chromosome number 2, chromosome number 3. So here you can find chromosome number 1 of maternal, chromosome number 1 of paternal over here. This is what I was talking about. Now, because these two belongs to the same chromosome, now these two is called as homologous chromosome. So if you can see over here, paternal chromosome and the maternal chromosome, and when they replicate, they become sister chromatids. Right? So that's a sister chromatid, that's a sister chromatid. If I'm talking about chromosome number 12 over there, so for example, if this is a chromosome number 12 over there, now this, when they undergo replication, they form sister chromatid. Otherwise, they are just chromatids. Now they become sister chromatids, or also called as chromosome number 12 or 13 or 14, whichever number you want to give over there. Now, a chromosome number 1 will never pair up with a chromosome number 6 or chromosome number 9. Now, chromosome number 1 and chromosome number 9 becomes non-homologous chromosome. But if it's the same chromosome, then you call them as homologous chromosome. So how the karyotyping is given. So you must have done this in your practical session. You must have stuck pictures of this chromosome in your practical classes in your records, where we give groupings to these chromosomes over here. So what are the groupings over here? So if you take chromosome number 1, 2, 3, chromosome number 1, 2, 3 are metacentric. Chromosome number 1 and 3 are metacentric. Chromosome number 2 is submetacentric. So under group A, chromosome number 1, 2, 3 are placed. Now the groups are A to G. A, B, C, D, E, F, G. So these are the chromosome groupings that you say. Now, when you're talking about chromosome, the group B chromosome, the group B under chromosome under group B chromosomes are 4 and 5. Now 4 and 5 are submetacentric. They are large and submetacentric. Why large? You can see. So these are large ones when compared to other chromosome over there. So they are large ones. Chromosome number 4 and 5 kept in group B. So these are submetacentric. Now you will say, ma'am, how are you able to identify and tell us that this is a metacentric, this is a submetacentric, and this is submetacentric? Now, if you can see the constriction, the primary constriction, the centromere position over here, the centromere position is dividing this chromosome into two equal arms over here. Now, that is the reason why I call them as metacentric. Now, if you can see this chromosome number 2 centromere is dividing the arms into two unequal arms. One is a short arm, one is a long arm. Now, the short arm is called as a P arm, the long arm is called as a Q arm over here. And that is the reason why we call it as submetacentric. And the same thing goes to the chromosome number 3, because it is having two equal arms, then you call it as metacentric. Now, if you're going to group B, because you have two unequal arms, P arm is small, Q arm is long, then you call it as submetacentric. Group C, chromosome number 6 to 12, all these come under group C because they all are of the same size, same length, whatever you call that, the length of the chromosome. If you can see the length of the chromosome, almost of the same height. So you are keeping chromosome number 6 to 12 in group C, and all are submetacentric chromosome. Then we are coming to group D. Now you can see a very peculiar thing happening over there. You can find there's a secondary constriction on the P arm and a satellite you can find over there. You can see the satellite over there. Now, wherever there's a satellite, then you call it as acrocentric chromosome. If you remember, the chromosomes, the P arm is very, very small and the Q arm is very, very long in acrocentric. And in acrocentric chromosome, you find satellites. So if you can see those round ones over there on top, these are called as acrocentric chromosome. Why? Because they're having satellite over there. Which chromosome? Chromosome number 13, 14, and 15, they come under acrocentric chromosome, that is under group D. Now, when you're coming to group E, all group E's are submetacentric. So one, let's say for example, chromosome number 16 is metacentric and chromosome number 17 and 18 are submetacentric. Then you must be asking, why are we keeping metacentric over here? Why? Because the length is the same over here, and that is why they are grouped under E. But chromosome number 16 is metacentric, chromosome number 17 and 18 are submetacentric. Now, why you have to understand all these things is because if you have to read a lot about the genome, then all this is very important. The grouping is very important when you're talking about the genes that are present in each chromosome over there. Now, coming to group F, all are metacentric, that is 19 and 20 metacentric chromosome, and group G, 21 and 22, again you have an acrocentric chromosome. So 21, 22 are very, very small acrocentric chromosome. Now, coming to chromosome, the allosomes, the sex chromosomes, what you call it, is up there. X chromosome and the Y chromosome. X chromosome is always placed in group C over here. Why? Because this is submetacentric. So if you can see over here, 6 to 12 chromosomes are placed in the group C, and X chromosome is also placed in group C over here. Why? Because this is a same length of that of the C group, and that is why X chromosome is placed in group C, and this is submetacentric. Where do you find this Y chromosome? Y chromosome is very, very, very small, and they are grouped in G group. So they are very small, and G group is placed over here in the G group, that is Y chromosome. Very small chromosome that you find over there. So this is how you are going to group your chromosomes. And then when you're talking about, after grouping them, you will talk a little bit about this banding patterns over there. As I told you all, what are the banding patterns over here? The light color is euchromatin. The dark color over there is the heterochromatin. Euchromatin and heterochromatin. Heterochromatin does not participate in transcription, whereas in euchromatin helps in transcription or production of your proteins, your enzymes, your hormones, or all is because of this particular euchromatin that we have in our DNA. Okay, now the banding patterns, if you can see, each chromosome has different banding, and the chromosome number 1, which is longest one, will have maximum number of banding patterns. You can also check it in your textbook. You can just open your textbook to find out this unit 2. In unit 2, we are talking about the chromosomal banding patterns over there. So depending on the condensation period, as they're condensing, now during condensation, it might have 800, and later on when it is condensing even more, it will have 400 bands. When it is condensing even more, you will be having only 200 bands. But again, when you stretch, the banding again becomes more and more. Say, for example, when this is condensed, then this becomes a single band patterns over there, or these two can become band having only 2 banding patterns, and when you're expanding them, they will have more banding patterns over there. Now, how do you differentiate and how do you get to know the banding patterns in the chromosome? Now, this is again a beautiful experiment that was done over here with the help of G banding. There are different type of banding techniques. You might be asked a question, discuss the common banding techniques used in the chromosome karyotype. So what you're going to do is, you must have done this in your practicals as well. Again, I'll just go through what exactly it is. What is a G banding? G banding is nothing but the G stands for GIMSA. GIMSA is nothing but that particular dye that you use to stain the chromosome. Okay. Now, when you use GIMSA, the GIMSA stains one dark and one light over here. So the dark patterns, the dark bands are rich in AT, and the light patterns are rich in GC. Now, what is this AT, GC? Adenine, thymine, guanine, cytosine. So AT rich means these are all heterochromatin. GC rich are all euchromatin. Light bands, GC rich, euchromatin. Dark bands, AT rich, heterochromatin. I'm sure you've understood what exactly I'm talking about. AT, heterochromatin, does not involve in transcription. GC, euchromatin, involves in transcription. Okay. So light bands are GC rich and AT rich are dark bands. Dark bands are heterochromatin. Light bands are euchromatin. If you still have a doubt, you can just again ask me once again. Okay. Now, this is one more type of a banding, reverse banding or R banding. R banding is nothing but a reverse banding. What is this reverse banding? Reverse of G banding is R banding. So what is the reverse of G banding? Now, this AT rich all now becomes light bands and GC becomes dark bands. So just a reverse of AT and GC banding of G banding over there. Now, coming to Q banding is also known as quinacrine dyes, which is fluorescent in nature. So, for example, if you have a fluorescent microscope, then you can use this quinacrine banding. Instead of this contrast black and white color, you can also have a fluorescent color to be very specific what exactly you all are searching for. So, quinacrine banding. Now, there is also called as a C banding, which is also called as centromere banding or constitutive banding over there. Stains only the constitutive heterochromatin. What is constitutive heterochromatin? Very, very tight bands. So, for example, centromeres, wherever you have the primary constriction and secondary constriction, you will be able to find out in this C banding. There's one more called NOR banding, nucleolar organizing region. That is the secondary banding patterns wherever you want to find out in the acrocentric chromosome where the secondary constrictions are present. Then you have this NOR banding. Then you also have one more called T banding, telomerase banding. So you want to find out if the telomerase of that particular chromosome is safe or as it decreased as we age, as it decreased. So for that you also have T banding technique called telomerase banding patterns over there. So that was about banding techniques over here. Now coming to how we are going to karyotype the chromosome. Say for example, you go to the genetic counseling because nowadays the parents, if the age of the parents is too high, they will have this problem of genetic problem in the progeny, because as they are aging, as a woman ages, the eggs are also aging, and Because of that, there can be some genetic abnormalities in the child. So to rule out that, people go to the genetic counseling, give their blood samples, or give the amniocentesis, the growing fluid in the womb, to find out if the baby's karyotype is normal or abnormal. Or for even for us also, if we say there's something problem with us, then you can go for karyotyping to find out if everything is fine or not. So what we do? So what are the samples that are needed for karyotyping? First thing is that you have to understand what is the samples that you need over here. First thing is you can take the peripheral blood. Peripheral blood is taken out from the veins, and then you can go for centrifugation, take out the WBCs, and you can do this karyotyping. The second thing is you can also find the crime scene. There'll be very less samples over there. Even the root tip, hair tip is enough for us to take out the samples over there. The saliva is enough to take out the samples over there. The bone marrow is enough to take out the samples over there to find out karyotyping. So these are some basic information of how you can take out the samples over there. So mostly, if you're going to do amniocentesis also, when you want to talk about any abnormalities in the growing fetus, then amniocentesis can also be done. What is amniocentesis? The fluid that is present in the womb. You can take out the fluid because child's DNA is present in that womb. And this amniocentesis is done only within 8 weeks of gestation. Further than that, it is a little, not 8 weeks, 16 weeks, sorry. 16 weeks. Later on, it is very difficult because even the amount of DNA is also decreased over there, and for abortions to occur also, it is very difficult. So before, from say around 10 weeks to 16 weeks, you can get this amniocentesis done over there. Okay, now coming to karyotyping. So what is this karyotyping? If you can see this picture over there, the karyotype over here. Now, all the chromosomes are same up till 22. And here in the allosome over here, you can find there is one X chromosome and one Y chromosome. So 46, how you write the karyotyping? 46, why? Because if you count the numbers, you will have 46 number of chromosomes, one from each parent, one from each parent over here, that is one from the maternal and one from the paternal over there. So if you count them, you have 46 number of chromosomes over here, and the X and Y chromosome. So you're going to differentiate the 44 are the chromosomes of the somatic cells, and 45 and 46 are the allosomes or the germ cells, which will differentiate whether it is a male or a female. Now, in this particular picture, if you see, you have one X chromosome and you have one Y chromosome. That means X and Y chromosome. Then this particular karyotype is a normal male. And for example, if you have two X's over there instead of Y chromosome, if you have two X's over here, then you call it as, how do you write the karyotype over here? 46, XX, and you call it as a female. So that is a normal female chromosome. That is the karyotype of normal female and normal male. 46, XX, 46, XY, you call it as normal male or a normal female. So something like when you go to the blood, when you give some blood samples for diagnosis, positive or negative, so something like that. When you go for a karyotyping, this typing is given over there. 46, XX means normal female, 46, XY means normal male. But for example, if there is some syndrome or if there is chromosomal abnormality that is found, say for example, in this picture, if you see over here, all are having 2, but chromosome number 21 has 3, one extra chromosome is present at chromosome number 21. So here you will find only 2 over here, but here it is 3 over there. So that means one copy is extra over here. So how do you karyotype this? When you count the whole chromosome, you will have 47. Why 47? Because normal is 46. Because there is one extra chromosome, you write it as 47. If you count also, you will get a 47 over here. And whether it is a male or a female, if you find over here, it is having 2 X's over here. So comma XX, because you have 2X. Why? There is no Y because already you have 2X. That means this is a female, XX, comma plus. That is plus means addition. What is addition over here? Chromosome number 21 is extra over here. So you write 1 plus and 21. Why you write as plus 21? Because you have to tell which chromosome is extra. Why which chromosome is extra? Because instead of 46, you wrote it as 47. So that means one chromosome is extra. Now, which one is extra is what you're going to talk about as plus 21. Why 21? Because chromosome number 21 is extra over here. Then you write this as chromosome number 21 is extra. 47, comma, XX, because it is having XX over here, comma, which chromosome is extra? Plus 21. Now, this particular syndrome is called as Down syndrome. Why? Because chromosome number 21 is extra. The name of that particular syndrome is called as Down syndrome. Why Down syndrome? Because the scientist named Down discovered this particular syndrome. So on his name, you call this as Down syndrome, presence of extra chromosome number 21. Now, for example, if there is this, you have two X chromosome. If one X chromosome is missing over here, that means you will have only 45, comma, XO. One X is present, one X is minus over there. You don't know whether minus is X or a Y. So XO over there. So you call it as Turner syndrome. So in the same way, Klinefelter syndrome, if there is one X, two XX is already present. If you have one Y also present over here, that means one extra over there, 47, comma, XXY, comma, XXY, because you're already written the sex chromosome over there as extra over here. So that is why it is called as Klinefelter syndrome. We'll talk about these characters in the coming slides over there. As of now, I hope you have understood this. I want you all to understand what is fish technique over here. This is one more topic that is very much important in your question. They might ask you to describe fish over here. What is fish? Fish is not the fish that we eat over here in the market, but the full form of fish is fluorescence in situ hybridization. The fish stands for fluorescence in situ hybridization technique. That is, you're going to use a fluorescent probe and you're going to hybridize them into your DNA. That is called as fluorescence in situ hybridization. Now, this technique is becoming very, very popular. Why? Because if you want to identify any abnormalities in the chromosome, say, for example, if there is a gene deletion, gene duplication, complete chromosome missing, then this in situ hybridization comes into effect over here. So you use a probes over there. The probes are always fluorescent induced. You tag some fluorescent chemicals to the particular probe that you have prepared by primer technology over there. And then you're going to prepare them ready to identify the genetic disorders. How you do this? You take out the samples. The sample is fixed onto the slide. Then you add an alkali over there. The first thing is you wash the DNA with some detergent, and then you denature the DNA. DNA is double helical structure. Now, this double structure is separated over there. Once they are separated, once this double helical structure is separated, the probes that you're going to use is going to get tagged on to that specific probe where you're going to go and make them join over there to find out what is the anomaly that is present over here. So how you identify this over here? So examples over here, if you can see over here. Now, this is a particular cell over here. And this particular cell, if you want to find out if there is any problem over here. So, for example, if this is a Down syndrome over here, in the Down syndrome, what I told you all, what is the karyotype of the Down syndrome? Plus 21. Now, usually you should have only two chromosomes, one maternal and one paternal over here. But here, instead of one maternal and one paternal, there is one more extra 21 over here. So that is why you write it as 47XX plus 21. So three chromosomes, chromosome number 21 is present over here, which is being identified by in situ hybridization or the fish technology. So this under the fluorescent microscope, you can find them. So this is one type of identifying with the help of fish technology. Then there is one more over here. For example, if there is a gene deletion or duplication over here, for example, here I'm talking about chromosome number 11 over here because the number 11 is written over here. Chromosome number 11, if I'm going to talk about this particular gene that is present on Q arm over here, Q arm, why I'm talking this as Q arm, it is also written over there, 11Q 21.3 over there, right? So 21.3, that means chromosome number 11 on the Q arm, I am at second place 1.3. So this is the place where I'm talking about. Now, if this is present over here, everything is normal. Now, when you view it under the microscope with the fish technology, you can find all the colors that are present over there, the red color and the yellow color that is present, the red and the yellow, very simple yellow over there, and a green also is present over here. So a red, a yellow, and a green that is present over here. That is normal over here. Now, for example, if this green has if this red is lost to another chromosome, if you can see over here, now the normal one is green and red over there. But here what is happening? Only green is present. The red is lost to some other chromosome over there. It has lost over there. So that means over here, this particular gene, red color gene, is missing over here. If that gene is missing, that means there is a problem in the humans over there. So if that is a problem, that will create some particular type of diseases. So for example, it is not for sure. For example, if that particular gene, which is going to produce insulin, is deleted, that means the person is not able to produce, or they are not tolerant to that particular sugar level, and that is the reason why they have this diabetes. To find out this, they will also have this fish technology. So if you can see over here, one particular chromosome is normal, which is having this red and yellow as this one over here. But the other one that has lost it, you will have one red at one place and green at one place. So the red has gone to one chromosome, the green has gone to, it is staying in one chromosome over there. So here they are separated. So that means there is some problem in the breakup of the example that is being given over here at so this is 11Q 21.3 over there. Now coming to amplification, if there is an amplification or say for example duplication of the particular gene that has been found out. So for example over here, now because there is 2 that is written over there on top of the chromosome, that means I'm talking about chromosome number 2. The position of the genes that we are talking about, this is 2p24.3 over here, and there is one more 2q11.2 over there. So these are particular 2 important genes that are present. And when you undergo a fish technology over there, everything is normal. Green is present, red is normal. So each chromosome having one over there. So one from maternal, one from paternal, one from maternal, one from paternal, normal. But what happens over here, this particular gene has got amplified. Amplified means they have become excess. Duplication has occurred, and that particular gene has become excess. Now, if they are becoming excess, that means there is a problem over there, because anything in excess is also dangerous over there. If you have excess amount of proteins or excess amount of hormones, they can lead to some different problems over there. So how you find out what happened to that particular gene? Again, fish technology. Now you can see over here, this red color is amplified too many times over there. Instead of 2 over here, there are many number of red targets that are present over here. That means the fish technology tells how much of amplification has occurred to that particular gene of interest that we are talking about. So this is fish technology. So I hope you have understood till now what we were talking about, and also what are the chromosomes over there. What I want you all to do is, I'll just finish off this particular slide, and then we'll wind up in the next class over here. What I want all to show is, when I'm talking about the chromosomes over here, we are so close to our human cousins over here. For example, the chimpanzees, the orangutan, or the gorilla over here. We share with the chimpanzees, we share about 98.8% of the DNA over here. That is why we call them as cousins over here. But who is the very closest cousin to us is the bonobos. I don't know how many of you all must have heard of this bonobos. The bonobos are close, very, very close, at 99% of the genome we share with the bonobos over here. So these are the amount of genes that we share among them. And I want you all to understand the difference in the chromosome numbers over here, and this is Ophioglossum. Ophioglossum is a plant which is this is a plant, Ophioglossum, a fern-like plant over here. How many number of chromosome it has? It has 1260. That is the highest number of chromosomes you find in this Ophioglossum. Now, this Ophioglossum is very, very important medicinal point of view. Why? Because they extract olive ointment from them for medicinal use, which can be also used for medicinal purpose over there. Now, this was all about chromosomes, the karyotyping, the number of chromosomes that you found, and the different numbers, the banding patterns, how the chromosome is packed up, and all those that you find over there. So in the next class, we'll talk about the abnormalities, and in between, if you all have any doubts, you can come to me. You can ask me questions regarding the same. And if there's anything that you want to chat and ask for any doubts, you can just chat, message, or you can also unmute and talk to me. Girls, I find three of them over there. Sadat, Shravani, and Umeria Syed. Any doubts, students?
Right now, no doubts, ma'am. We will refer to the textbooks and we will come to you.
Okay, thank you so much. And you can also, I hope you people have a group, your WhatsApp group. Are you all there?
Yes, yes, ma'am.
If possible, you can just add my number into your group, so that if there is any doubts you people want to ask, you can just message to me, and I am there at any given time. You can message me. Sadat, if you can just take out my, I hope that's a name, because I can see only
Yeah,
that.
yeah, ma'am. Yeah, ma'am.
Okay.
Just
If you can
please
just take out.
text your number, no, ma'am. Yeah, text your number in the chat.
Aha, yeah,
So
yeah, that's the same number. Which one? 9490937422, right?
okay. Yeah, yeah.
Yeah,
I
yeah,
will add you in the group.
yeah. You can just add me, and then we can keep exchanging any information, or if you
Yeah.
want any extra classes also regarding the same,
Okay.
I am there.
Sure.
And I would like all the whole class to participate in this particular class, because this class is going to be very, very interesting. And if you all are going to miss, then I'm sure you
I'll
all are going to.
inform the other students also, ma'am. I'll inform
Yes,
the other
please do
students.
that. Please do that.
Ma'am,
Yeah.
will you please repeat your number?
9490
9490
9374
9374
double two.
double two.
Yeah.
Okay, ma'am. Thank you, ma'am. Thank you
I'm
so much.
Padmini over here, right?
Okay, ma'am. Sure.
Okay, nice meeting you all, students. Meet
Thank
you all in
you.
the next class. Tomorrow's class, I just wanted to inform you all. Tomorrow's class, I'm a little bit busy. I will take my class on 19th. The next class is on 19th, right? Next Sunday.
Okay. Yes, ma'am.
So I've requested Anita madam to take two classes. Tomorrow she will take two classes, one her class and one my class. And on 19th, I'll be taking her class and mine class together. Okay?
Oh,
At the given time itself. Okay, ma'am.
okay, ma'am. Okay,
Yeah.
ma'am. Thanks, ma'am. Bye.